In the heart of life's complexity, there's a dance unseen yet profound. Witness the epic spectacle of cell division, mitosis. A mesmerizing journey that shapes life itself, captured in every intricate detail. Join us as we delve into the realm of creation and witness the miracle of growth. Welcome to the wondrous world of mitosis, where the magic of life unfolds before your eyes. First, let's dive into the world of mitosis and meiosis, two essential processes in biology. Discover the key differences that make them unique and crucial in the study of life sciences. The cell cycle comprises three main phases: G1, where the cell prepares for division, S, where DNA replicates, and G2, the final preparation phase. Additionally, we have the core mitosis phases: prophase, metaphase, anaphase, and telophase. Unlike mitosis, meiosis produces genetically diverse cells used in sexual reproduction. It involves two sequential divisions, meiosis I and meiosis II, resulting in four non-identical haploid daughter cells. Where do mitosis and meiosis occur in the human body? Mitosis mainly takes place in somatic cells throughout the body, ensuring tissue growth and repair. In contrast, meiosis occurs specifically in germ cells within the reproductive organs, testes in males and ovaries in females. These unique locations enable mitosis to maintain body functions. Next, we'll explore meiosis and garlic root cells in the lab. To initiate the experiment, secure an onion or garlic bulb in a beaker with toothpicks. Once the roots reach a length of approximately 2 to 3 centimeters, it is optimizable to trim 1 centimeter of the root tips and place them in a small vial containing carnoise fixative solution. This solution preserves the DNA in the root tip cells. Leave the roots in the fixative for approximately 48 hours. If you intend to store the root tips for future use, you can transfer them to another vial containing 70% ethanol. In any case, extract several root tips from either the carnoise fluid or ethanol and place them onto a watch glass containing 1N-hydrochloric acid. The acid's role is to gently soften the cell walls and weaken cellular connections, making it simpler to squash the root material at a later stage. Immerse the root tips in the acid for approximately 2 minutes. Afterward, rinse the root tips a couple of times in distilled water. Next, transfer the roots to a watch glass containing either acetocarmine or gentian violet stain. These stains are known for their excellent ability to color the nuclear material of the root cells in a deep red hue. Allow the root tips to remain in the stain for approximately 5 to 10 minutes. Subsequently, transfer the root tips onto a clean glass slide that contains a small droplet of water, employing a sharp blade or scalpel. Eliminate about 1 millimeter of the root tips and dispose of the excess material. The active cell division occurs in the very tips of the roots, which are the essential regions needed for mitotic experiments. Carefully place a cover slip over the root tips, ensuring there are no air bubbles. Gently tap the cover slip with the blunt end of forceps or a pencil rubber until the root tips are evenly flattened between the slide and the cover slip. A correctly squashed slide will display a faint, cloudy pink to nearly colorless appearance. Your root tip slide is now prepared for microscopic examination. This is what a finely flattened root tip looks like under a 10x microscope. The cells of the root tips are evenly spread out with minimal overlapping. If the root tips are not adequately squashed, you can repeat the squashing process to achieve better cell distribution. Scan the slide to identify cells displaying mitotic division stages under low magnification and then proceed to observe them under higher magnification. Microphotographs are captured to identify various mitosis phases. Further analysis includes measuring cell diameters and assessing mitotic activity to gain insights. Then we delve into a practical illustration of the consequences. When mitosis malfunctions, cancer can result from genetic alterations that disrupt the cell cycle, predominantly affecting mitosis. Protoncogenes and tumor suppressor genes, when mutated, can promote and cannot restrain cell division. DNA repair gene mutations lead to genomic instability. Genetic changes impacting mitosis can lead to tumor formation. Tumors can be benign, staying localized and less harmful, or malignant, spreading and posing more significant health risks. Cancer can spread throughout the body via the bloodstream, a process known as metastasis. Malignant cancer cells can invade blood vessels and travel to distant organs, forming new tumors. This ability to metastasize makes malignant cancers more dangerous. Understanding the mechanisms behind cancer is crucial for diagnosing and treating this disease. 1/3 of people today will develop cancer at some point in their lives. Cancer now has more than 100 different types identified. Among these, breast and lung cancer stand out the most severe forms, life-threatening and high-frequency. But our ability to fight cancer is improving. Since just the 1990s, cancer mortality rates have dropped more than 26% in the U.S., and more than 2 million lives have been saved. As treatments and methods of early detection improve, scientists and the public better understand cancer. Hope remains in the fight against the disease. Recently, significant progress has been made in cancer research. Studying mitosis is crucial for understanding cell division and disease. It informs the development of treatments, diagnostics, and prevention strategies, benefiting individuals and society.